Hello dear learners. In our today's class, we will learn about the antigenic determinants on B cell and T cell epitopes. So, in short, in this video, we have to learn about the epitopes and the specificities of these epitopes to be recognized by the B cell and the T cell. So, first of all, let us understand what is the meaning of epitope. An epitope is a region which is present on the antigen. Antigen is any foreign substance. On this antigen there is a specific region which we are calling as the epitope, and this is the epitope which is responsible for the activation of the immune response in the host organism. This is that region which can be detected by the antibodies. Means the antibodies are basically trying to recognize the antigen through this epitope. So when we talk about these epitopes, these are the antigenic determinants, means the determinants of the antigen, which are immunologically active regions, means they have the capability to induce the immune response in the host organism. They are very specific because these are antigen specific membrane receptors and are specific to the particular antigen on which they are present. Now these epitopes are known to bind with the B cell and T cell receptors so that the immune responses can be generated. In this video, we have to talk about the specific epitopes properties which can be useful for the B cell recognition and the specific epitope properties which are useful for the T cell recognition. So first of all, let's talk about that what should be the properties of the epitopes if they have to be recognized by the B cells. Means what should be the properties of B cell epitopes. So the first important property of the B cell epitope is that the epitope must be exposed onto the surface of the antigen. Now there is a slight difference between the recognition pattern of B cell and T cell, and that you should all know. What happens is that when I talk about B cell, suppose this is the epitope of the B cell, so the B cell through its receptor will recognize the whole of this epitope along with this whole antigen. Means the soluble form of antigen will be recognized by the B cell. So this is your B cell, this is your B cell receptor which is in the form of immunoglobulin, and this is your antigen. So in order to be recognized by the B cell, or in order to be having this affinity with B cell, the particular epitope must be exposed onto the surface of B cell so that B cell can easily recognize the particular epitope. So that is what it is written here, and also that these epitopes should have the complementary shapes with the B cell receptors so that the maximum possible non-covalent interactions could occur. Always remember that the epitope and the antibody binds with the non-covalent interactions which operate only over shorter distances. So now if the non-covalent interactions are there which are the weak bonds, then how to strengthen them? To strengthen them, one thing which is possible is that the close fit shape can be possible. If this is like this, then there can be many non-covalent interactions through which this bonding can be stabilized. So this is what the first property of the B cell epitope must be. That is, the epitope must be exposed to the outer surface of antigen because the B cell receptor can recognize only the full and the soluble form of antigen. Let's move on to the another property. The another property which is important is the size of the epitope which is present on the antigen. Now always remember that when I talk about the size of epitope, that epitope which is comparatively smaller in size will be compatible with the B cell receptor, but that epitope which is comparatively larger in size will not be compatible with the B cell receptor because this small epitope can easily fit into the groove, but the larger epitope will not be able to fix into the groove. So the second important property is that the epitope must be compatible to the groove of the BCR. Large epitopes will cover the superficial surface area, but the smaller ones can fit deeply inside the groove. Also remember that the typical amino acid sequences for these epitopes must be around 15 to 22 amino acids, because this is that optimal amino acid sequences through which there could be a good interaction surface of approximately 750 angstrom square. This much surface area can easily be covered, and that is very well good for the uh, bonding between the epitope and the antigen. Let's move on to the next important part. The next important property of the epitope is that the B cell and the T cell epitopes can contain the sequential and the non-sequential amino acids. Let us take this example here. If I say that the B cell epitope is in a way that, hmm, let's change the color. Suppose this is the normal amino acids which are there in the epitope, and then these are those particularly epitopic amino acids, and after that again normal. So there can be this case where all these are present in the sequence. So this kind of epitope is the sequential epitope. Then there can be another also. For example, I have small here sequences, then the protein is folded in a way that the remaining sequences come here, and then again it continues like this. So here what happens is that here we should have the interactions. Now these kind of interactions and these kind of sequences are non-sequential because they are not in the continuous order. They are randomly arranged, but the conformations have brought them closer. So the non-sequential epitopes can also exist, and the sequential epitopes can also exist for the BCR. If we are providing heat, if we are trying to denature the protein, still the sequential epitopes can be recognized because the sequence will never be broken. But here if we are denaturing it, so it means that here these sequences can also break, and that time when this epitope is divided into fragments, the recognition cannot take place. So always remember the sequential epitopes even after denaturation. Now when I talk about the term denaturation, it means the unfolding of the protein molecule, not the breaking of bonds or not the breaking of the chain. So denaturation will lead to the loss of epitopicity of the non-sequential chains, but in the sequential chains, their property of acting as the determinants would not be lost. So this is another important property of the B cell and T cell epitopes. So of the B cell epitopes very specifically. There is also one of the particular examples here. We have taken here two examples. One of them the hen egg white lysozyme, and another one is here myoglobin whale sperm or sperm whale myoglobin. So these are the two specific epitopes. Now please remember that when I talk about the sperm whale myoglobin, this is a kind of antigen which has the sequential epitopes. Sequential epitopes. But when I talk about the another one, that is hen egg white lysozyme, it is non-sequential epitope. So now you can clearly see that the bonding is in a way that the 64th amino acid has some kind of interactions with the 80th amino acid, and when this is happening only then this sequence is retained, and this conformation should be retained. If the conformation is retained, that time we can say that the epitope is functional. But if we open the loop, if we try to open the loop, that time the epitope becomes non-functional. So that is also the important thing which you should know. Moving on to the next property is the conformational dependence, which I already discussed above also, but you should be knowing that if the epitopes are particularly have the proper conformation, that time they bind more effectively, they have the better binding. But if the conformation is not proper, that time the binding is also not very good. Another important part is that the B cell epitopes are generally present or located in the flexible region of the immunogen. Now this is the particular immunogen, and this is suppose this region which I mark it in, let's say red, this region, it is very flexible, means it can be easily fitting into the narrow spaces. So if that epitope is present here, suppose if that epitope is present here, that time it is a better epitope, because we are not very sure about the B cell receptor and its width. So if this region is flexible, it will be able to fit into the width of that B cell. So that is the important property, that if the epitope is also present in that flexible region, it can also change its shape and accordingly fit into the B cell receptor. And more the better flexibility, uh, more the binding between the epitope and the B cell receptor. So these flexible regions are also sometimes referred to as the mobile regions of the antigen. Moving on to the next, we talk about the complexity. The complex proteins, they contain multiple overlapping B cell epitopes, and some of them become immunodominant. So immunodominancy is also one of the important properties, which means that if suppose there are on a particular immunogen there are four epitopes present, E1, E2, E3 and E4. Suppose E2 is that epitope, if it binds to the B cell receptor, it is bringing the highest amount of immune response, or if it is able to generate the highest amount of immune response in the host body, that means it is the immunodominant epitope. So if the epitope is immunodominant, it is a preferable epitope for the BCR. So these are the characteristic features and these are the characteristics of the B cell epitopes. Now we should discuss about the characteristics of the T cell epitopes. So if I talk about the characteristics of the T cell epitopes, these are also quite simple. See, first important point for T cell epitopes I should write here is that the epitopes required for the T cell are or should be linear. Now, as I was talking about that B cell can recognize the whole of the soluble form of antigen, the T cells cannot do that. For the T cells, the condition changes. First of all, that particular antigen or the immunogen has to be phagocytosed by the antigen presenting cells. After that, it has to be lyased inside either through the proteasome or through the endosomes, and then they have to be represented either through MHC1 or through MHC2 onto the surface. The whole of this pathway has been already covered in the previous videos. You can have a look on them also. So now here this only the peptide of that antigen will be present here, that is antigenic peptide, which will be shown off by MHC1 or MHC2 molecule. Now this antigenic peptide is actually the epitope only, and this epitope here is linear type. So always remember that the epitopes of the T cells are always linear and the processed form of the antigens. The conformational epitopes or the globular epitopes or any other kind of epitopes won't be working here. The more complex the epitope is, the lesser it would be presented by the major histocompatibility complex, and then the lesser it would be recognized by the T cells. So this is one of the most important properties of the epitopes for the T cells. Second important thing which I should write here only, that is these particular epitopes which we are talking about is has one thing very important, that is the epitope should be internal in origin. Suppose this is your particular antigen, then the epitopic sequence which has to be recognized by T cells should be internal into the antigen, means should be present around here. Means it can also be, the plus point is it can also be hydrophobic, but in the case of B cells it should always be exposed, means there the compulsion was that the epitope must be hydrophilic in nature. Why it can be inside? Because it will, when it will be processed, this part, suppose if it is green in color, this green part will eventually be exposed out, and that part will then be further expressed by either MHC1 or MHC2. So the second important property is that the epitope should be internal in origin. Thirdly, there is specific sequences which should be there in the epitope. First of all, the epitope must be in the form of linear peptide, and secondly, to be recognized by MHC1, the sequence should be only 8 to 10 amino acids long, and in order to be recognized by MHC2, the sequence should be not more than 13 to 18 amino acids long. And this what I have covered in my previous videos also. Next important thing, for the T cell to be activated, the trimolecular complex should be formed. Now what is the meaning of trimolecular complex? The trimolecular complex here is between the epitope, between the MHC molecule, and between the T cell receptor. I can also show you a very good diagram here which has been given in the book only, that is this one. So here you can see how the antigen presenting cell APC is there. This is exposing its MHC. Suppose it being the class 2 MHC molecule. This is the antigenic peptide. Then here is the T cell receptor. So this particular trimolecular complex should be there, consisting of class 2 MHC, consisting of the antigenic peptide, and consisting of the T cell receptors. Then in support of that, this is CD4 here. Why CD4? Because it is the determinant, or it is the cluster of differentiation for the class 2 MHC. That is for the this particular TCR, that is T helper cell. So this is also one of the important thing that we should know. And then last thing, last but not the least, the antigenic processing. There are already different pathways for the processing of antigens, and we specifically know those pathways. See, if we talk about the MHC1, then the antigen processing should be done endogenously. That is, the epitope must be generated endogenously. If I talk about the MHC class 2, then the pathway should be exogenous, or the epitope must be generated exogenously. So these are the characteristic features of B cell and T cell epitopes. That is when we talk about the antigenic determinants. Now in the next video we are going to talk about that what should be the characteristics, what are the characteristics of antibodies. That is, we'll talk about the isotypic, allotypic and idiotypic antibodies.